Today I am going to explain about seed germination. The resumption of active growth of the embryo resulting in rupture of a seed coat and emergence of a young plant is called as germination. There are three stages of germination. One is pre-germination, second is germination, and third one is post-germination. In case of pre-germination, first the rehydration takes place, that is imbibition of water. RNA and protein synthesis is stimulated. Increased metabolism, that is increased respiration, takes place. Later the hydrolysis of reserve food material by the enzymes will be done. Then change in cell ultrastructure will be seen. Then induction of cell division and cell growth will be seen. In case of germination, the rupture of seed coat, emergence of seedling, usually radicle will be the first. Then post germination, controlled growth of root and shoot axis, and then controlled transport of material from food stores to growing axis is seen. Then senescence or aging, senescing of the food storage tissues will be seen. There are three phases of seed germination. One is imbibition phase, second is latent phase, and exponential phase growth. In case of imbibition phase, water is absorbed by the seed, which activate the gibberellic acid synthesis. This increased gibberellic acid synthesis will in turn lead to increase in DNA synthesis, which led to the RNA synthesis or transcription, which finally led to the production of amylase enzymes or other hormones or other enzymes also, which this amylase enzyme will degrade the starch, convert into sugar. This sugar enter inside the epicotyle, which led to the growth and germination of the seed. Latent phase is further divided into phase 1, 2 and 3. In phase 1, rapid uptake of water takes place and the onset of mRNA synthesis will start. Then the onset of starch degradation takes place. Then glycolysis and fermentation of starch hydrolysis, which led to the cell wall biosynthesis, non-functional protein degradation, mitochondrial assembly and amino acid biosynthesis. Then in the phase 2, that is TCA cycle, where in this phase 2 coleoptile emergence will be seen. Then starch biosynthesis, stocking protein degradation, and then aerobic respiration will be seen in the phase 3. In the exponential growth phase, the seed coat will break up and then the sugar produced by the aerobic respiration will be consumed by the embryo, and epicotyle and radicle and the hypocotyle will start to grow, and that will lead to the germination of the seed. There are two types of germination. One is hypogeal germination and another one is epigeal germination. In case of hypogeal germination, the cotyledon is present inside the ground level after germination of the seed. That's why it is called as hypogeal germination. Hypo means inside the ground level. So hypogeal means inside the ground level. And this type of hypogeal germination is seen in monocot plants. In case of epigeal germination, the cotyledon will come above the ground level after the germination. That's why it is called as epigeal. Epigeal means above the ground. So this type of germination is seen in case of dicots. Requirements of germination. First is the light. Generally seed require darkness to germinate, but sometimes it requires light. Photoblastic. If a seed responds to light for germination, named as a photoblastic. Three categories of photoblastic seeds are there. One is positive photoblastic, that is lettuce or tobacco are some of the examples, which require exposure of sunlight. Whereas negative photoblastic, here example is onion, lily, amaranthus, and they do not require exposed sunlight. Non-photoblastic, here non-exposure of light is needed. Phytochrome is a plant pigment found in cytoplasm that sense the presence of light, that is red light. Phytochrome absorbed light in two interconvertible form. PR is metabolically inactive and absorb red light, that is 660 nanometer. PFR is metabolically active and get transformed into PR. The later promote the germination, that is PR will promote the germination, and other phytochrome control the process of the plant. PFR revert back to PR after absorbing far red light at 730 nanometer. This is the light spectrum where you can see that the nanometer for red light is from 650 to 760. This is the wavelength in nanometer. In function of phytochrome system will be explained in this picture. Here the inactive form, that is PR, that is phytochrome which absorb the red light at 660 nanometer, will be converted into an active form which is PFR. That will lead to a photomorphogenesis. Whereas when this PFR when it is in active form, that lead to a dark light, and it will be produced into, it will be converted into a inactive form. Now this PFR will absorb the far red light at 730 nanometer and converted into PFR, which is an inactive form. So usually in this table you can see that whenever a red light is given to the seed, it will germinate. When you give a red light plus far red light, no germination will be seen. In the same way, if we give red light, far red light, and then the red light, germination will be seen. In the same way, if you see here, whenever the last light which will be given in red form, then germination will be seen. Whenever there is a last treatment will be far red light, then there will be a no germination. So we have to remember in this way, when there is a red light, seed germination takes place. When there is a far red light at the light treatment, then no germination will be seen. To germinate, seed must take in water. Uptake of water by seed is called imbibition. The imbibition lead to swelling and breaking the seed coat. Next is oxygen. Aeration. Oxygen is required initially for metabolic activity. Breakdown of stored food for energy, radical emergence from the seed coat with aeration. Temperature. Most seed go through cold period before germination. It helps prevent seeds from sprouting as soon as they mature. Most common annual vegetable have optimal germination temperature between 75 to 90 degree Fahrenheit, that is 24 to 32 degree centigrade. Temperature must be optimum. If the temperature is very high, then seed will emerge very fast, and even weather. Traditional kharif crop are sown in June and July after receipt of monsoon southwest monsoon, whereas a rabi crop during the October, November. Quiescence. Seeds are not provided with adequate condition like water, temperature, or air for germination. That is seeds in storage form. Dormancy. Seeds remain ungerminable even though provided with suitable condition. Then it is called as dormancy.